Previously on cardiophysiology, we were able to give a good description of the cardiovascular system. We discussed the types of circulation, and we were also able to explain the anatomy of the heart. If you are yet to see the video, make sure you do so through the link on the description of this video. In this particular video, we are going to have a good look at the myocardium. First of all, let us run through some of the properties that can only be seen on the cardiac muscles. Although the myocardium contains contractile elements such as actin and myosin, which can also be seen in skeletal muscle, it still has some fissures that can only be seen in them but not seen in skeletal muscle. Instead of having longitudinal fasciculus of muscle cells as seen in skeletal muscles, the cardiac muscle cell is branched, and the branches of cardiac muscle cells make end-to-end contact with branches of the adjacent cells. Cardiac muscle have special intercellular connections. The sarcolemma of the cardiac muscle is thinner than that of the skeletal muscle, and because of this, it makes it possible for substrate and gases to be able to diffuse into the cardiac cells. There are two groups of intercellular connections of the cardiac muscle. There are connections that strengthen the junctions between the muscle cells, and there are connections that permit cell-to-cell conduction of impulse. In connection that permit cell-to-cell conduction of impulse, there are gap junctions along the longitudinal surface of the adjacent cells, through which ions flow from one cell to the other. This feature is the reason for the syncytial behavior of the myocardium, which we will discuss later on. As for the connections that strengthens the junction between the muscle cells, there are presence of structures called intercalated discs. These intercalated discs are due to the extensive folding of the cell membranes at the ends of those connecting cells, merging with Z zones of several myofibrils on either sides. Other strong bonds such as tight junctions and desmosomes do exist. Another special feature that is only peculiar to the cardiac muscle is that the muscle cells have one nucleus that is oval in shape and found in the center, unlike the skeletal muscle that has more than one nuclei and are located on the periphery of the muscle cell. The cardiac muscle cells also exhibit aerobic metabolic activities due to the presence of large numbers of mitochondria that are found crowded between the myofibrils. Another important feature that is very unique to the cardiac muscle is that their mitochondria and sarcoplasmic reticulum are very rich in complex lipids like phosphatids and some cholesterol, which are extremely important for their enzymatic activities. Cardiac muscle is similar to skeletal muscle because it also contains myoglobin, just like the skeletal muscle, and this myoglobin does the work of hemoglobin in the blood, that is, the transport of oxygen. Let's look at some metabolic activities that occur in the myocardium. In the myocardium, the energy that is needed for the contractions of the cardiac muscle are almost gotten entirely through aerobic metabolism, unlike skeletal muscle. And because of this, the heart cannot be sustained on anaerobic metabolism for a long time, and therefore cannot be allowed to incur oxygen debt. The reason for this is because energy gotten under anaerobic condition has a very insignificant 1% of the total energy need, and which is not even enough to sustain the contraction of the ventricles. The substrates that the cardiac muscles use to carry out their metabolic activities are fatty acids, which provides about 60% of the energy. Another substrate is carbohydrate, that is lactate and glucose, which provides 35% of the energy, and ketones as well as amino acids, which provide the remaining 5% of the energy. Although only a small quantity of amino acids are metabolized, the myocardium contains higher concentration of aspartate aminotransferase enzyme more than any other tissue in the body. And this enzyme gets to be released into the blood circulation whenever there is death or damage of any part of the myocardium, during prolonged starvation and in untreated diabetes mellitus. More fat is metabolized. In the cardiac muscle, the rate of metabolism is directly proportional to the peak tension generated during cardiac contraction, and to some degree to the length of the time the tension is maintained. The efficiency of normal heart is 20 to 25%. Let's look at the electrical properties of the myocardium. We will start with the very first, which is action potential. The action potential in cardiac muscle is very prolonged. The total duration of action potential in the ventricle is about 250 milliseconds compared with the duration of action potential in the skeletal muscle or in the nerve, which is 2 to 4 milliseconds. The reason for this prolonged duration of action potential in the myocardium is due to a delay in the repolarization phase, which manifests as a plateau in the action potential, unlike the spike potential seen in skeletal muscle or nerve. There are differences in shape, ionic mechanism, and duration of action potential in various parts of the heart. Let's look at the various action potential in various parts of the heart. But firstly, we are starting with the action potentials in the ventricles. The action potential in the ventricle have 5 phases, that is from phase 0 to phase 4. Phase 0 is also known as phase of rapid depolarization with overshoot. In this phase, the resting membrane potential is rapidly dropped from minus 90 mV to 0 mV and then reverses in polarity so that the inside is positive, which is about plus 25 mV relative to the outside. The reason for this is because of the opening of voltage-gated sodium ions with a rapid influx of sodium ion and some calcium ion. The gates open up when the resting membrane potential drops to about minus 60 mV. This potential at which the gate opens is known as the threshold potential. We should be conversant with voltage gating. What voltage gating means is that the gate, which is ion channels, open or closes depending on the voltage across the membrane. Phase 1 is also known as phase of initial rapid repolarization. In this phase, from the new positive membrane potential, rapid decline to 0 mV occurs, and this is due to the closure of sodium ion channels and influx of chlorine ion. Phase 0 and phase 1 correspond to the QRX wave of the ECG. Phase 2 is also known as the plateau phase. In this phase, the membrane potential remains stable for about 150 milliseconds. This is due to the prolonged opening of the voltage-gated calcium ion channel, allowing a slow influx of calcium ion. This phase corresponds with the ST segment of the ECG. Phase 3 is also known as rapid repolarization phase. Rapid repolarization returns membrane potential to the resting level. What causes this is due to the closure of calcium ion channel and opening of both voltage and ligand-gated potassium ion channels, which causes potassium ion efflux. This phase lasts for 50 milliseconds and corresponds to the T wave of the ECG. It coincides with the end of systole. The last phase, which is phase 4, is the phase in which potassium channel close due to no further change in membrane potential, and sodium ion potassium ion pump restores sodium ion and potassium ion to their original positions on the either side of the cell membrane. In our next video, we will continue with the action potentials in the atria. Subscribe, like and share. See you next time.